So I want to tell you a little bit today about work that's been going on in my lab over the last few years on the influence of the cell lineage on the outcome of a herpes viral infection. In particular on how lineage affects the genetic program of the virus, how that change in the genetic program is manifested as a difference in signaling, and how that signaling difference turns into something that has pathogenetic consequences and may even have therapeutic consequences. And before I go on any further, you can tell from the amount of gray hair on the speaker that when I say we, I don't mean me. As the Talmud says, the work of the righteous is done by others. And the other in this case is Henry Chang, who's a graduate of UC Berkeley's PhD program and has been postdocing with me initially at UCSF and came with me to Novartis a couple of years ago. And everything I'm going to show you today is the product of Henry's work. Okay, a little primer. We may need the lights down a bit for this on KS. So KS is a tumor of endothelial cells. Endothelial cells are the cells that line vessels in the vascular system. I'm oversimplifying here grotesquely because KS is a quite complicated neoplasm. But in normal hosts, people with a normal immune system, KS is an indolent process. This is a tumor that people die with but not of. It's largely confined to the skin. For peculiar reasons, it's confined to the skin of the lower extremities. The lesions are highly vascular, so they're even red to the naked eye. And this is what KS looks like in a healthy immunocompetent person, in which it's a relatively rare tumor. But of course, in HIV infection with immunodeficiency, and this is also true of patients with organ transplants, KS has a much more malign countenance. It's very widespread on the skin, can be disfiguring, and it can also occur or spread to organs, especially the lungs. This is what's responsible for fatalities in KS, is visceral involvement in patients with HIV. Now, one of the questions that has been bedeviling pathologists in the study of KS is what kind of endothelia does KS arise from? Does it arise from endothelial cells that line blood vessels, vascular endothelium, or does it come from vessels of the lymphatic system, which are drainage systems that connect up to the venous system and drain tissue fluids back into the venous system? These are two separate lineages within endothelia. They can be differentiated by their markers. And it's been a debate going on back and forth for a long time whether KS is derived from lymphatic endothelium or vascular endothelium. And I'll come back to this in a minute. Just hold that in your mind. Now, the causative agent of KS is caused by a herpes virus, KSHV. And like all herpes viruses, these are huge DNA viruses. KSHV is 165 kb genome. There's over 100 open reading frames. And like all herpes viruses, textbooks say that there are two genetic programs that herpes viruses engage in. A latency program in which the genome persists in a cryptic form, largely in the nucleus, with highly restricted gene expression. In KSHV latency, most of the latent gene expression comes from this small collection of genes shown in the box here, just a handful of genes. The cell survives. No virus is produced during the latent state. If the right stimuli impinge on a latently infected cell, it can flip into the lytic program, which is more like the program that you're accustomed to think about in virology. All the viral genes are expressed in a temporally regulated cascade. The cell gets sick and dies and releases large numbers of virus particles into the medium. Okay. Now, in KSHV infection, by the way, at least in cell culture, latency is the default pathway. If you put virus onto most cell lines, they enter the cryptic or latent stage. And you have to treat them with certain stimuli in order to reactivate the lytic infection. Now, a few years ago, the whole debate about whether KS derives from lymphatic or vascular endothelium, which was at the time favoring lymphatic endothelium because KS never arises in organs like the brain that have no lymphatic system. And KS endothelial cells have many lymphatic markers in them. That whole debate was upset by the discovery that when you infect lymphatic endothelium in culture, the markers change to be more like vascular endothelium. And when you infect lymphatic endothelium in culture, the markers change to being more like vascular endothelium. So this is called reprogramming of lineage markers, and what it indicates is that you can't use these markers in a biopsy of an infected individual to say anything about the lineage from which they derive. So what was once a settled pathologic argument is now back in debate. Okay. Now, that debate was not something we were thinking about very much when we began this work. The work I'm going to show you that Henry's carried out over the last few years was actually prompted by a clinical paper that had to do with the treatment of the immunodeficient form of KS. In this case, solid organ transplant recipients who get KS and have this more adverse prognosis were shown to successfully respond to treatment with rapamycin. Now, prior to this date, the way you treated transplant-associated KS was to try to lighten up on immunosuppression, okay? With the idea being that if the people are less immunosuppressed, they'll control the tumor better. And there's a lot of truth to that. And in fact, the idea of trying rapamycin in these patients is due to the fact that rapamycin is a much less potent immunosuppressive than some of the calcineurin inhibitors like cyclosporin. And in fact, this was the first really effective treatment for post-transplant KS. If you switch them to rapamycin, they in general can maintain their kidney allograft function and have a striking regression of KS. But the question really was, is this because of the reduction in immunosuppression, or is it because of other signaling activities of rapamycin, which is known to signal through the mTOR pathway, which is an antagonism of which is known to be antiproliferative? Okay, so this is the fundamental question. Okay, and this is the question that got us into the work that I'm going to show you today. Okay, now just to remind you that rapamycin acts by inhibiting mTOR signaling. mTOR stands for mammalian target of rapamycin. mTOR signaling is a signaling cascade that involves integrating signals from growth factors or the energy charge to a pathway that converges on mTOR, a multi-subunit complex, in this case mTOR1 complex 1, that acts to upregulate translation and cell proliferation by hitting a bunch of targets, including S6 kinase that phosphorylates ribosomal protein S6 and ups the translation level, 4EBP, which when phosphorylated liberates EIF4E in its active form. So these are all factors that upregulate translation and growth. Okay? And mTOR is regulated by a gap called TSC, which in turn is classically regulated by phosphorylation through the PI3 kinase and AKT pathway, AKT being the direct kinase that phosphorylates TSC. Okay, so this whole pathway is under negative regulation of TSC, and it's released from that typically by AKT phosphorylation. Okay. All right, so that's the mTOR pathway, and the observation that rapamycin was active in transplant KS led us to examine the state of the mTOR pathway in endothelial cells that were infected with KSHV. Okay, so now KS occurs in the skin, and the targets are dermal endothelium. And as I said before, these can either be coming from dermal lymphatics or dermal blood vessels, and you can actually prepare primary cultures of microvascular endothelium that's either lymphatic or vascular in origin by sorting them according to certain markers. I won't go into that, but these can even be bought commercially now. Primary cells directly from human skin. Okay, both lineages can be infected by KSHV. That was already known. So we infected them with a recombinant version of KSHV that is marked with GFP in latency and RFP expression in the lytic cycle, so we can tell latent from lytic infection. This virus also carries a puromycin resistance marker, so you can select it. Okay. So what we're going to show you is the result of infecting lymphatic endothelial cells, LECs, or blood endothelial cells, BECs, with this recombinant KSHV219 that's marked in this way. Okay. We select stable, latently infected cells with puromycin, and then we examine their growth, signaling, gene expression, and rapamycin sensitivity. Okay. Very straightforward protocol. All right. So the first thing that we noticed was there was a very striking difference, depending on the lineage, about the state of activation of the mTOR pathway. that if you infect LECs with the 219 virus, mTOR is activated, but not blood endothelial cells. So lymphatic endothelial, but not blood endothelial cells. So here's the experiment, mock infected, infected, and we're looking at a whole series of markers for the state of activation of the mTOR pathway. You can see that in terms of S6 kinase, it undergoes strong activation when you infect LECs, and as a result, S6 protein gets phosphorylated, and these are all markers for mTOR pathway activation. I'll say that for EBP1, which is a widely known substrate of mTOR signaling, behaves very variably in cultured cells. This is a known feature. The most reliable way to mark mTOR pathway activation in cells is with S6 kinase and S6 phosphorylation. Okay. Blood endothelial cells, on the other hand, display no changes in S6 kinase or S6 phosphorylation before or after infection. Okay. Now, next we asked about the state of AKT phosphorylation, because this is the typical canonical pathway that leads to activation. And we got our first big surprise, which is that there was no activation of AKT, no phosphorylation of the relevant serine on AKT after infection in lymphatic endothelia, in which we know the pathway is activated. So that was a big surprise. This is not happening at the level of AKT, but somewhere below that. And while we were doing this work a few years ago, a second pathway for activating mTOR became known in the signaling field, and that is that activation of the RAS, RAF, MEK pathway, which is very common in cancers, is also known to impinge, have crosstalk with the mTOR pathway, largely through a protein called RISC, which is a substrate of ERK. And RISC can carry out several phosphorylations on mTOR itself and on TSC that release this negative inhibition and allow activation of the pathway downstream of AKT. So we examined that pathway in infected lymphatic endothelia. And what we found is that this pathway is activated in infected endothelium in a lineage-specific way. The lymphatic endothelia show strong upregulation of RISC phosphorylation, of ERK phosphorylation, and interestingly enough, there isn't any activation of the pathway above ERK. The MEK and RAF are not activated, so this is occurring not through growth factor stimulation like this, but some intracellular process that is acting at the level of ERK and RISC. I'll come back to that in a second. But I also remind you of one other thing, or want to point out one other thing, which is that ERK has two isoforms, 1 and 2, and only ERK2 is activated in the context of infected lymphatic endothelia. And none of this, of course, is going on in the blood endothelial lineage. All right. So how does KSHV do this? And fortunately for us, there were some clues in the viral literature about this, because a man named Fanshu Zhu, who's now at Florida State, was at Penn before, had discovered that there's a virally encoded protein, the product of a gene called OR45, that can activate S6 kinase activity. And it binds S6 kinase and activates it. Now, he hadn't made the connection to mTOR activation in that work, but there was one other thing that was interesting in his earlier papers, which is that even though it does not bind directly to ERK and RISC are in a complex, and when that complex existed, ERK2 was selectively activated by the expression of OR45, which was exactly what we were seeing in the infected cells. So we had a strong clue that OR45 might have something to do with this, and I'll show you data that affirms that now. So first of all, if you take OR45 and put it in a retroviral vector and use it to transduce lymphatic endothelial cells, you can phenocopy the entire infection with regard to the mTOR signaling pathway. So you can see phospho-RISC, very strongly activated, selective activation of the ERK2 isoform, and downstream activation of the S6 kinase pathway. And you can also see that TSC2, the negative regulator of the pathway, undergoes phosphorylation at the site 1798 that is known to be hit by RISC. Okay? And in fact, when you use OR45 alone, there is no activation of AKT phosphorylation, just as we saw in the infected cell. So this gave us a strong clue that OR45 was likely the agent involved. And in fact, we can demonstrate that by going back to the infected lymphatic endothelium and use siRNA to selectively knock down OR45 in the context of that infection. And when you do that, first of all, you get a very good knockdown of OR45. You can see that the risk activation, phospho-risk, is knocked down. That selective activation of the ERK2 isoform goes away, and so does downstream signaling in the mTOR pathway. So clearly then, OR45 is the gene product in latently infected cells that lymphatic endothelial cells that's responsible for the mTOR phenotype. Okay, now here's the thing. OR45 is not supposed to be a latent gene product of KSHV. Okay? OR45, according to the canonical understanding, is a lytic gene product. It's only on in cells that are going to make virus. And that was a profoundly paradoxical thing, and suggested that there was something very different going on in lymphatic endothelium, in this lineage of endothelium that was not going on in the other lineage, the blood lineage. And to get a better feel for that, we carried out an analysis of the entire viral transcriptome using a tiling array that tiles across the entire viral genome. And when we did that, we got a very striking result, which is shown here. So this is a little complicated, but along the horizontal plane are individual oligos that represent the entire viral genome. They're single-stranded oligos, so that the Watson strand is shown on top and the Crick strand is shown on the bottom. And when you do a lytic infection, a proper lytic infection, and you see that virtually every gene in the viral genome is expressed. That's what this control lane is. This is a standard in vitro cell line that has been treated with 4-ball esters and is induced to lytic replication. It's making large amounts of virus, 10 to the 7th copies per mil in the medium. Okay. This is what a conventional latent infection looks like in most cells. You can see that there's very strong attenuation. In fact, this is the same cell line before you add the inducer. You can see that all of the gene expression is coming from one region, that boxed region I had shown you earlier. This is classic latency. Okay? If you look in blood endothelial cells, you see more or less the same pattern. Actually, interestingly enough, there's an additional cluster of transcripts that we haven't yet fully characterized. They're antisense to a bunch of interesting things in the viral genome, including a collection of virally encoded microRNAs. And they are on in these blood endothelial cells, although they're not on in the standard in vitro line that we use of a tumor line. So maybe they're interesting and they need to be looked at more. But overall, this is a pattern that is really fairly typical of canonical latency. But when we look at infected lymphatic endothelium, these are just replicate experiments, we see a very dramatically different pattern. Something that looks a lot more like lytic infection, although there are many lytic genes that are not on here, that are on in lytic infection. But there's just a plethora of genes that are annotated as lytic genes that are on here. And this is really not in accord with the textbook understanding of what herpes viruses are supposed to do. And again, the effect is lineage-specific. It's lymphatic endothelia and not vascular endothelium in which we observe this phenotype. Okay. If you look at by Western blotting, we don't have antibodies to all those proteins, but for the ones we do, KB-ZIP, OR45, RTA, these are known lytic gene products. Kaposin B is a protein that's made at extraordinarily low levels in latency, but strongly upregulated during lytic infection. It's also on. And the canonical latency marker is also, ELANA is also on. ELANA is on in blood endothelial cells, but all these markers are on in lymphatic endothelial cells. So this isn't just a transcriptional phenomenon. This is going on at the protein level. As the OR45 siRNA experiment also affirmed in the signaling context. Okay, now there are two possible interpretations you could make of this. One is that there's actually just a lot of lytically infected cells in that culture. The other is that every single cell has a program that's different from latency analytic reactivation. And first of all, and there are two arguments that I can produce here that suggest that it's the latter. This is going on in every single cell. First of all, these cells were selected in puromycin, and they're growing as a mass culture. That is not compatible with the idea that every single cell in the culture is making virus, or that a large number of cells are making virus. When we assay virus in the supernatant, we do see a small amount of virus there, 10 to the 3 copies per mil. This is 3 to 4 logs less than you would expect from a massively lytically infected culture. So if, in fact, the burst size per cell is the same, then only 1 cell in 1,000 to 1 cell in 10,000 in the lytic cycle here, canonically understood. Also, I remind you that there were a lot of genes that we didn't see expression on in the tiling array that are known to be on an lytic infection. So even the tiling array is suggesting that's not the case. But lastly, when we stain by IFA for proteins that are known to be lytic-specific, what we discover is if you compare the number of GFP-positive cells to the number of ORF59 positive cells, in this case, that we're staining for, over 60% of the GFP-positive cells are staining by immunofluorescence. Now, that is probably just a difference between the sensitivity of immunofluorescence and the sensitivity of direct staining for GFP. But at a maximum, half the cells that are growing must be in this alternative state, and probably all of them are. Okay. So what's the phenotypic consequence of that deregulation of gene expression? With regard to the thing we began with, the therapeutic question, how does rapamycin act here? So we asked, what is the growth phenotype of cells that are from the lymphatic lineage when they're infected, and what happens to them when you treat them with rapamycin? So in blue are the uninfected cells, in red are the infected cells. These are lymphatics, these are blood endothelial cells. You can see that in blue, the uninfected LECs grow normally, and they grow normally in the presence of rapamycin. If you infect the LECs, they grow more slowly, but something very dramatic happens when you treat them with rapamycin, which is their cell number appears to go down. They look like they're dying. And that is not the case for blood endothelial cells. Blood endothelial cells, even when they're uninfected, are more sensitive to rapamycin growth inhibition, but they don't die. And infection makes no difference to them. So this is what it looks like, and I'll just call your attention to over here. Here are LECs treated with rapamycin, and here are LECs that are infected and then treated with rapamycin. And you can see there's a lot of killing going on that dish. Whereas blood endothelial cells, whether they're uninfected or infected, are not particularly affected by the presence of rapamycin. Okay. And this killing is apoptotic in origin. If the lights were down here completely, but if you look closely, you can see this is tunnel staining with LECs that are infected before and after treatment with rapamycin. You can see a whole lot of cell loss, and you can see all these red dots everywhere, which is tunnel staining indicating that these cells are undergoing an apoptotic program. So not only have the cells activated mTOR signaling, they have become dependent on it. If you withdraw it by poisoning it with rapamycin, the cells die. This is signaling addiction, as the cancer biologists like to call it. These cells have become addicted to the mTOR pathway. And I'll close by just telling you something this is more evidence that that's true. I'll close by just showing you what we've learned about that addiction process. We don't understand it fully, but we do know this, that ORF45 alone, which is responsible for the signaling activation, is not responsible for the addiction, okay? If you take retrovirus that transduced cells with a retroviral vector for OR45, these are lymphatic endothelia that have been transduced with OR45. They have signaling activation, but when you treat them with rapamycin, they don't undergo apoptosis as judged by PARP cleavage or caspase activation. By contrast, if you take a lymphatic endothelium and infect it with the whole virus, and then knock down OR45, you diminish that apoptotic response as judged by PARP and caspase activation. So what this suggests is that OR45 is responsible for upregulating the signaling pathway, but it's not responsible for the addiction. The addiction is a secondary event that happens downstream of OR45, either due to one or more of those many other viral genes that's on, or formally speaking, even a host gene that might have been turned on by the pathway. That's what's responsible for the addiction. It's actually going to be quite interesting to try to figure out what that biochemistry is, but we don't understand that yet. So I'll just close by by trying to tell you what the main take-home messages are here. First of all, endothelial lineage strongly affects KSHV infection. It affects it at the level of the transcriptional program, that LECs display a very deregulated form of transcription that's not been seen before in any other herpes viral infection, and it is a violation of the sort of dualism that textbooks teach about latency and lytic reactivation. That upregulation includes OR45, which activates mTOR signaling, and secondary events downstream of that cause the cells to be dependent on mTOR. So what does this mean for KS? I think what it might mean, now, first of all, let's be honest. This is all going on in primary cell culture. This is nothing like a real human tumor. We're not reproducing all of KS here. But it does suggest that there is a direct effect, anti-proliferative effect of rapamycin in infected cells. And that suggests that perhaps the drug could be used for classical KS or AIDS-related KS, and not just the transplant-associated form, where you're trying to also benefit from the reduction in immunosuppression. That's not proven by any of this, but it does suggest a clinical experiment that we hope someone will do. And finally, the effectiveness of rapamycin in KS, in human KS, might be considered an argument that perhaps lymphatic endothelial cells are the target of KS, or perhaps at least they're playing a very important role in the maintenance of a KS tumor. I don't want to insist on that because this is all in vitro work, and primary cells are not the same thing as a living cell. But it is something that's formally possible and is going to need a lot more work to determine. I don't want to insist on that, but I think what's really important here is that lineage affects the viral transcription program, and many interesting phenotypic things flow from that. Thank you.
Hi, Dan.
Hola.
I was wondering, so your data would suggest that there's something maybe fundamentally different about how viral genomes are getting epigenetically modified during the latent programs of LEC and BEC, and have you compared the modification state of the viral genome in those two cells and looked at maybe some of the cellular factors?
Yeah, so this is a big area of ongoing work. Presumably, well, we know that all viral transcription and latency comes from a chromatinized plasmid in the nucleus, and so we're now looking at the state of that epigenetic plasmid, the epigenetic marks along the entire viral genome using ChIP-seq, and this is work that's going on in the lab now. There's no conclusion yet, but I'm sure there are going to be very different markers there, or at least I'm hoping that there will be. Otherwise, this is going to be a much more complicated problem than we thought.
Do you have evidence in vivo, in lesions, that there is virus production going on at a low level?
Yeah. The last time anybody looked at this was over a decade ago when we looked at it with Ashley Haas by in situ hybridization. At the time, we had very few markers for latency and lytic reactivation. In fact, the first markers we cloned for latency and lytic reactivation were applied directly to KS tumors back then. But they don't differentiate between, and what they show is that a lot of cells are latently infected in the classical sense, but there are other cells, up to 5% of the cells in the population, that had lytic gene markers in them. We don't know now, in retrospect, whether they're in this program or they're in true latency. They appear to be the minority, but not a small minority, in severely immunodeficient hosts. But this all needs to be re-examined now. We need to identify which genes are not on in this program that are on in lytic infection, so that those programs can be differentiated. And now we need to make antibodies to those things and go back with immunohistochemistry to and that's a very big task, but that's where the game is right now.
I'm curious whether you have looked at the substrate, the downstream substrate of S6K, and have you looked at the...
Yeah, we looked at S6 and S6 phosphorylation, and they are in accord with the activation state of S6 kinase.
Have you?
That was on many of the slides.
Have you looked at the actin cytoskeleton proteins?
No,